Hello everyone, I am Sita Sirisha, working as a postdoc in Dr. Lee's lab at University of North Texas Health Science Center, Fort Worth. Our lab mainly studies CRISPR-Cas proteins computationally using machine learning and traditional chemoinformatic techniques. In this work, as the title says, we identify family-specific features of CRISPR-Cas proteins utilizing machine learning models built on a complete spectrum of protein features. CRISPR and its associated Cas proteins together form the CRISPR-Cas technology, which is basically an innate defense mechanism of bacteria. The technology has now been transformed into a gene editing tool that promises gene-level corrections in many genetic diseases. Cas, which is the most important component of the system, suffers from some inherent limitations like specificity and off-target effect. To address these limitations, it's important to know the features that govern this Cas family. Here we try to identify the family-specific features of two most popular Cas families, which is Cas9 and Cas12. With an objective to identify family-specific features, we built random forest binary classifiers to differentiate Cas9 and Cas12 families from the non-Cas proteins respectively. We also built multi-class classifiers to differentiate between Cas12, 9, and non-Cas proteins. Using this methodology shown in Figure 1, we were able to identify several family-specific features that are unique to the Cas9 and the Cas12 proteins. The methodology starts with feature generation using iFeature, which is an open-source Python package, followed by training data split, followed by feature selection using a five-fold incorporated Burda-Shaf algorithm that works based upon the game theory approach. The identified features are then used in random forest model building, training, and tuning. The developed models were then evaluated on the test set followed by independent data sets. We repeated our procedure 15 times to cover the variance across our dataset. Table 1 shows the various databases like UniProt, PDB, and Interpret, which were used to collect the Cas and the non-Cas proteins of our dataset. For feature generation, we use the iFeature package, which calculates various structural, physicochemical, and evolutionary features of the proteins. The study used a total of 13,495 features. We evaluated our models using 6 evaluation matrices such as accuracy, precision, recall, F1, AUC, and specificity. This was also followed by an independent set prediction. Coming to the results, as seen from the prediction results in tables 2 and 3, all the 3 models performed extremely well on the test and independent datasets. On the test set, the binary models achieved a high accuracy of more than 96%. On the independent sets, Cas12 models achieved an accuracy of more than 92%, while the Cas9 binary models achieved an accuracy of close to 95%. Likewise, the multi-class models also showed a high F1 score of more than 97% on the independent dataset. Further, we went on to identify the top 10 important features for Cas9 and Cas12 families. As you can see in Figure 2, the Cas12 family's top 10 features include features like normalized van der Waals volume, polarizability, charge, and other composition, distribution, and transition kind of features. Interestingly, the top 10 features are majorly dominated by 4 quasi-sequence order features, namely Schneider-Lag 2, 5, 10, and 17. Therefore, the Cas12 family is dominated by CTD and quasi-sequence order kind of features. Likewise, the Cas9 top 10 features are mainly predominated by amino acid composition category of features like the dipeptide, 5-peptide descriptors, which include descriptors like PC1, PP, XC1, etc. Mainly, we observe that 4 tripeptide composition features, namely PWN, PYY, HHA, and DHI, seem to predominate the top 10 features of the Cas9 family. A further multiple sequence alignment analysis showed that these tripeptides were found to be conserved in all the bacterial Cas9 species available from UniProt. Figure 4 shows how the 4 tripeptides were seen to be consistently higher in the Cas9 protein as compared to the Cas12 family. Upon mapping the 4 tripeptides on the widely used Streptococcus pyogenes Cas9 structure, seen in Figure 5, we saw them to be present in the catalytically important domains like REC3, HNH, and RUNC. Among these, the tripeptides DHI and HHA are known in the literature to be involved in the catalytic DNA cleavage activity of Cas9 protein. Mutation studies also showed that the tripeptide PYY and PWN are involved in the DNA binding activity of Cas9 proteins. They also play a role in improving specificity and reducing off-target activity of Cas9. Coming to the conclusions, we found that binary and multi-class models developed using the complete spectrum of protein features yielded a very high accuracy and F1 scores close to 95% and 98% on their respective independent datasets. We noticed that quasi-sequence order and CTD features predominated Cas12 family, while the Cas9 was predominated by the amino acid composition, especially tripeptide kind of features. Quasi-sequence order is vital for protein folding, while the TPC kind of features is important for the cleavage and other critical Cas9 functionality. Mutation studies showed that the tripeptide PYY and PWN are important for increased specificity, reduced off-target activity, and DNA cleavage activities. Identified features will also go down in future to serve as a guideline to design new Cas proteins with improved properties. I would like to thank the Department of Pharmaceutical Sciences at University of North Texas Health Science Center for giving me this opportunity. Thank you.